As researchers transition to animal-free testing, in vitro human cell culture models are a sustainable, effective, and reliable solution. With the Millicell cell culture insert, you can create biologically relevant skin models. In this video, we will show you how to generate the organotypic air-liquid interface, or ALI, 3D skin cultures. This serves as an in vitro model of the human epidermis and can be used in applications for cosmetic testing to clinical screening. Here is a diagram of the steps to generate an ALI skin model. In the first step of generating this model, we will create stratification of the skin. Mix collagen and fibroblast cells and place on top of the membrane. Collagen is very sticky and can solidify quickly. The best method is to add the mixture to the middle of the insert while ensuring the tip does not touch the membrane. This ensures an even collagen bed. Polymerize the collagen bed for 30 minutes in the tissue culture incubator. After solidifying the collagen, add in the primary cells. Now, place the plate into an incubator. After the polymerization, add the keratinocytes into the apical side. Add the media into the basolateral side. Submerge the culture for 2 additional days in growth media. More details on media exchange and respective volumes can be found on our website. On day 4, we will change the media to the 3D Grow differentiation medium. Remove the apical media. It is important that you do not add any liquid to expose cells to the air. Then, exchange the basolateral media into the 3D Grow medium. The volume will depend on your plate height. Ensure the media in the bottom or basolateral side is lower than the collagen bed. Now, to prepare to section and stain the membranes, fix with a standard fixative solution for tissue processing. We will be using 4% paraformaldehyde for 15 minutes. Flip the insert upside down and use a number 11 blade to excise the membrane. Gently hold the membrane with forceps, noting the side of the membrane with cells. For sectioning such as H&E staining, place the membrane in a microfuge tube for further post-processing. In following these steps, you can expect to see these 3 separate layers in your culture, ensuring confidence when performing downstream assays. You can also stain for Ki67 and filaggrin to confirm the proliferation and differentiation of the skin model. Downstream assays can be performed on the skin model, such as permeability assays, tear readings, drug screens, and wound healing. For more information, please contact your account manager or visit our website.